I thought I'd go through that. To do that, I think properly I need to kind of give a little bit of background on what the drug is and kind of how the disease we're focusing on is really manifest, because that plays into the design that we started. So Tracon, we're focused really on one lead product, which is an antibody to endocrine called TRC105. And just to put it in perspective, it targets a pathway of VEGF resistance. And so our clinical development plan is really taking our drug TRC105, combining with VEGF inhibitors. And we're doing that across multiple indications, but our initial phase 3 is in angiosarcoma, which is a very rare orphan indication that we'll talk about in a little bit more detail. But we're also studying it in eye disease through a partnership with Cytel and also in fibrosis. So with respect to the clinical development plan, and we'll just focus, I think, today on TRC105, we're really focused on oncology as our lead therapeutic area. And within that, we're pretty broad in terms of development. We have a randomized study in renal cell carcinoma as we speak. There's a single-arm study in liver cancer and also a rare tumor type called GTN. But our main focus is the phase 3 angiosarcoma trial, which is now enrolling using this very unique adaptive enrichment design that we'll talk about. Just going to tell you a little bit about the target and why it's important and why we're focused on angiosarcoma initially. And that is, endoglin is a target that's expressed on proliferating endothelial cells. So it's expressed on the tumor vasculature of every solid tumor. And if you appreciate, for instance, on this slide, the liver cancer picture and the normal liver adjacent to that liver cancer, endoglin is not expressed on resting endothelial cells, but it's expressed when endothelial cells proliferate, as they do in cancer. So you see dense expression, for example, in the liver cancer on the tumor vasculature. Angiosarcoma is a unique tumor in the sense that it's endothelial cell malignancy. So you see really dense expression of endoglin, particularly on angiosarcoma. We think that's why we've seen particularly dramatic activity, namely complete responses when we dose TRC105 with VEGF inhibitor approved in that indication, which is pazopanib or Votrient, a drug made by Novartis, I should say marketed by Novartis, originally brought to market by GSK. So angiosarcoma is our initial lead indication based on the dense expression, but the key with the endoglin pathway is it really is an escape pathway that mediates VEGF resistance. So all our trials really are taking the antibody and building upon VEGF inhibitor. For example, in renal cell, we're building upon Pfizer's drug Enrichment. In angiosarcoma, because Votrient is the approved VEGF inhibitor there, we're building upon Votrient. And then the other play for this drug is AMD. You see dense expression on the choroidal vessels in AMD is shown in red at the bottom portion of the slide. Just one quick antibody side on how it actually works. Antibody binds to endoglin. Endoglin is part of a complex receptor dynamic. I think suffice it to say on this slide, by binding endoglin, we interrupt signal transduction through the activating ligand called bone morphogenic protein, or BMP9. We also directly mediate ADCC, or cytolysis of endoglin-expressing cells, as a second mechanism of action. And just to kind of summarize what we're doing clinically, we're focused on many indications, but we're doing development in a tiered fashion. We're focused initially on orphan drug indications. Angiosarcoma is the lead phase 3 indication. I mentioned we're in phase 2 studies, with the key randomized phase 2 data being renal cell data due end of this year, and that's combining the drug with Enrichment. That's Enrichment with or without TRC105, and that's an important data point expected end of this year. And then we're also in some larger indications, breast and lung cancer, just getting phase 1 studies moving forward there, with the anticipation of being potential indications across multiple therapeutic, or with the intent of being combined with VEGF inhibitors across multiple therapeutic indications in the oncology space. So now let's talk a little bit about angiosarcoma and about the clinical aspects of that disease, because the clinical aspects of the disease really were considered carefully in designing the phase 3 studies in an adaptive enrichment design. So first of all, angiosarcoma, you probably don't know much about it because it is a rare tumor, but it is a very morbid tumor. It's a lot like pancreatic cancer in terms of the available therapies. Now, most patients with angiosarcoma will receive first-line chemotherapy. Progression-free survival is about 4 to 5 months. Most people in the second-line setting, many will receive Votrient or pazopanib. And just to give you an idea how poorly effective Votrient is in this disease, what I've cataloged on this slide are 5 studies of single-agent VEGF TKIs in angiosarcoma. The most recent data, and this was actually even updated more recently this year, but in pazopanib's single-agent study of 30 patients, now it's been updated to 40 patients. 40 patients treated with operative angiosarcoma. There have been no complete responses in that study. The PFS, even at 40 patients, is still 3 months, and the overall survival is 9.9 months. So again, this is a very deadly tumor. You know, when you talk to sarcoma physicians and they say, you know, I've got a leiomyosarcoma, which is a soft smooth muscle tumor, you know, I just treat that patient today or I can treat it in 2 weeks. When you talk about treating an angiosarcoma, we need to start treatment tomorrow. I mean, it just has that impetus because it's so aggressive, you need to really treat aggressively and quickly because it really will progress that quickly. The other 4 studies, I won't go through in detail on this slide, but I think you can appreciate, you know, well over 100 patients, you know, there were 2 complete responses documented in this series of studies, 5 studies that treated well over 140 patients. Or see, well over 130 patients. So with that as the background, we had done a phase 2 study where we enrolled broad sarcoma subtypes. And we always were particularly focused on angiosarcoma because, again, it's an endothelial cell malignancy. Endothelial is an endothelial cell target. It should be highly responsive to therapy was the biologic hypothesis. We initially treated 9 patients, which has now been publicly disclosed. And interestingly, 3 of the patients had actually already progressed on Votrient as an initial or previous therapy. And we were impressed that we started seeing complete responses in these patients. 2 of the first 9 patients had complete responses. And not only were they complete responses, they became very durable complete responses, as I'll share with you on the next slide. Even in this set of 9 patients where 3 of them had already progressed on Votrient, we actually saw activity even in the patients that progressed on Votrient. One of them was a complete response, patient 1, for example. You know, patient 5 was actively progressing on Votrient, had a minor response to therapy. And then patient 7, even though they only stayed on therapy for about 6 months, they actually stayed on Votrient single-agent therapy for fewer than 6 months when it was the initial therapy. So even in the patient that failed Votrient, we saw signs of activity in this overall set of patients. This is a very refractory set of patients, but the overall PFS in this sample size was about 5.6 months, which compared favorably to that baseline 3.0 months with pazopanib single agent. Again, in the pazopanib single agent study, I should mention, none of them had seen pazopanib previously. So in our view, we saw much better activity in a much more refractory population, and what was particularly dramatic in this disease is complete responses. You just don't see those in this disease, given how aggressive it is. Just to kind of talk about 2 types of angiosarcoma, because that really plays into the statistical design for phase 3. About half of angiosarcoma is cutaneous disease, and about half is non-cutaneous or visceral disease. Cutaneous just meaning it starts anywhere on the skin. Non-cutaneous or visceral means it starts anywhere other than the skin. Typical patients get angiosarcoma. One typical patient would be a woman who's had breast cancer, who has had post-op radiation therapy. That's a risk factor for angiosarcoma. That, for example, is patient 1 shown on this slide. That's a woman who's had a mastectomy for breast cancer. She had post-op radiation therapy, and as a result, she developed a very diffuse angiosarcoma of her flank. Now, it may not project beautifully, but that red rash that's outlined or attempted to be outlined in black marker is angiosarcoma. The black marker is the physician's attempt to define the borders, but actually sometimes it's hard to define borders in angiosarcoma. But I think you can appreciate that's a pretty broad lesion. It's on the flank, it's going around to the back as well. And she developed a very nice complete response to therapy with Votrient plus TRC-105. She had progressed, to be clear, on Votrient and Taxol therapy previously. That has been impressive in the terms of seeing a CR, but what's really been impressive is the durability. You know, she's over 2 years of therapy. In fact, she's at the point where now we discuss, should we even stop therapy? You know, it's a hard choice to make because stopping therapy in a disease where if it recurs, what are you going to do next? Versus, you know, let's just continue therapy. She's tolerating it very well, and, you know, it's an important decision. But clearly a nice response in cutaneous angiosarcoma. Patient 2 also has cutaneous angiosarcoma. That's a patient who had a second risk factor, which is sun exposure for angiosarcoma. Now, clearly sun exposure, much more risk factor for melanoma, but also risk factor for angiosarcoma. That red rash, or looks like a sunburn of the face, that's angiosarcoma. You can kind of appreciate that raised border on the upper lip. It kind of gives you an idea. This is a raised lesion that's documented in angiosarcoma. The patient also responded very quickly to therapy, had a CR within 1 month of treatment. It's also been very durable. That's to be updated. That's going on at 24 months, so 2 years of a CR in that patient as well. Patient 6 is a patient who has visceral angiosarcoma. Now that's the forehead that's visualized on that picture, but that's a subcutaneous lesion that's tenting up the skin. And it may not project beautifully, but that's a very protuberant mass. And you can kind of see the fact that the ruler kind of moves across a circular lesion to give you an idea of the protuberance of the mass. nicely, that patient had it's not a complete response, but a very significant response. It's not completely flat, so we can't call that a CR, but it's clearly a very nice response. The person also had a cervical lymph node and also completely responded to therapy. So a nice response to therapy with visceral angiosarcoma. So I'm going to emphasize these two conditions, cutaneous and visceral. Again, 50-50 proportions roughly. If you look at the overall angiosarcoma literature, clearly cutaneous seems to be more responsive to therapy. And that seems to be, again, limited sample size seems to be what we're seeing also. We're seeing CRs in cutaneous disease. I think clearly there's activity in non-cutaneous, but it's possible that the response rate will be disparate between those two groups. So that was one consideration we thought about as we contemplated the phase 3 study. What we didn't want is, let's just say the drug's active in cutaneous patients. What we didn't want is to do a trial in both patients, have a negative data point when it works in cutaneous and it doesn't work in visceral as a potential outcome. So that was a key consideration in designing the phase 3 studies we'll talk about. Just in terms of how the timeline developed with the regulatory interactions, we were pretty aggressive moving forward in 2016, and it was the year of really positive regulatory interactions for this protocol. So we were able to achieve orphan drug designation in the U.S. in early 16, but as you likely know, that's simple. I mean, if you are less than 200,000 prevalence, you'll get orphan designation in the U.S. It's kind of a no-brainer. But in the EU, it's a little different. So the EU doesn't just require the prevalence to be less than 200,000 patients. For the EU to give you orphan designation, you have to show assumption of clinical benefit versus existing therapies. And the existing therapies, I said, for angiosarcoma are not very effective. First-line chemo is not effective. Second-line Votrient is not effective. And it was really the durable CRs that we were able to present to the EU that allowed them to say, yeah, there's an assumption of clinical benefit here. You're seeing CRs in a disease where you rarely see that, and they're durable that allowed us to receive that designation. That was really important because it allowed us access to the Special Protocol Assistance Program through the EMA, so we got a lot of interaction around the phase 3 adaptive design, and we were able to vet that through the EMA, and then also then vet it through the US FDA. So we did the EMA first through the Protocol Assistance meeting in August. We took that input, made some modifications, although they were very open to the adaptive design, very open. And we then moved to the end of phase 2 meeting with the FDA, who also were very open to the adaptive design we'll discuss. We had a successful meeting there and received the Special Protocol Assessment designation by end of the year. And now we've dosed the phase 3 study. The way the study plays out, we started dosing, I mentioned first quarter. We expect to do an interim analysis the first half of 2018 that will then inform us for the final analysis, which could be first half of 19 or potentially second half, depending on what the final sample size is, as we'll discuss talking about the study. So first of all, the overview of the study. So this is a randomized trial. And please, as I go through the slides, interrupt me for questions. I'm happy to answer questions. So a randomized study, initially 124 patient sample size. Patients either get Votrient single agent or they get Votrient plus TRC-105. Our goal is to increase PFS from 4 months to 7 months. And to do that, that's a hazard ratio of about 0.55. The initial sample size has over 80% power at a classic two-tailed alpha 0.05 to detect an improvement from 4 to 7 months. There are 2 strata in the trial. One is cutaneous versus visceral. I mentioned that there's clear evidence that that is prognostically relevant. Cutaneous does better than visceral. The second is the prior therapies. Now, interesting prior therapy discussion with the agencies, both in the US and in the EMA. I mentioned Votrient is not approved first line in angiosarcoma. It's used second line after chemo. But again, chemo is so poorly effective that both agencies were very open to actually enrolling treatment-naive patients into this trial. That's an interesting give in the sense that both agencies are willing to sign off on using a comparator drug in a sense off-label, right? Votrient's not approved first line. That they're allowing us to, in a sense, use it off-label in the first-line setting, which really takes into account the fact that chemotherapy, the standard first-line treatment option, is so poorly efficacious in this particular indication. So that, I think, will help us with accrual. And, investigators are obviously important to make that ask. They really felt strongly, there's no reason to just subject my patient to chemotherapy to get them on this trial. I'd rather just get them on the trial. So that is also important strata, though. We're stratifying first line versus second or third line patients into the study. So to be clear, patients can enter with 1 or 2 lines of chemotherapy, in addition come on at treatment naive. And our general view is about a third of patients will probably come on this trial as treatment naive. The key other give agency was that PFS is the endpoint, not OS. I mean, that's been pretty consistent in sarcoma, but it was still an important endpoint for us. Again, PFS is much shorter than OS in this disease, as you might expect, and also, I think, much easier to achieve. And you can reliably achieve PFS in sarcoma, and it is a reliable surrogate clinical benefit in this disease. Just general other eligibility criteria: age 12 or more, unresectable angiosarcoma, measurable disease by RECIST, no prior treatment with a VEGF inhibitor. I mentioned no more than 2 previous lines of therapy, and the ECOG performance status 0 or 1. This is an open label study, and that's because when you treat with TRC-105, you get a characteristic telangiectasia in the skin or small dilated veins to make it very clear the patient's receiving therapy. So in order to compensate for the fact that it's an open label study, we do have central independent blinded review with a company called Median. And we image every 6 weeks. Again, we expect PFS to be fairly short in this study. Unfortunately for the patients, we want to keep track of that with relatively frequent assessments. Any questions on the study design? I'll go into the adaptive design, but this is just kind of the general overall idea of the study.
2 questions.
Please, yeah.
The primary endpoint of PFS.
Yeah.
So this study design aimed for accelerated approval or full approval?
This is for full approval. Yeah, anticipation is 1 trial will be sufficient for full approval.
Based on PFS?
Based on PFS. Yeah, I think that plays into the fact that we're really in a rare tumor type. We're talking about 1,000 new cases a year. And I think when you are in a rare tumor type, and no one's ever done a phase 3 angiosarcoma, it's a rare tumor type, it's an orphan indication. It reminds me a little bit of when we were doing SUTENT, the SUTENT program, I felt like the agency wanted that drug approved as much as we did. Remember, when we were doing SUTENT, interferon alpha is how you treated kidney cancer. The worst drug ever invented. And interferon alpha is like giving yourself the flu every day, every week of the year. It's such a terrible drug. SUTENT changed the whole landscape of that disease, and the agency wanted that drug approved as much as we did. I think we get that same impression with these regulators. When you're in that true unmet need indication, no one else is there, you're really trying to advance their care, the regulators, I think, want you to succeed to the extent that they can do that. Yeah.
And I suppose this study is powered to show significant PFS.
Significant PFS, absolutely. 2-tailed alpha, 0.05. The power is a little bit above 80%. We'll go through the power calculations on a separate slide, but over 80% power at a standard 2-tailed alpha, 0.05, to show improvement from 4.0 to 7.3 months as the baseline considerations. Okay?
Thank you.
Yeah, no, great question. Please keep coming, yeah.
PFS based on central review.
PFS based on central review, absolutely.
What do you mean by confirmed by central review?
Right, no, you're right. It is by central review. Yeah, you're right. Don't need that word confirmed. Well said. Yeah. So just to formally go through the objectives, PFS is the key primary objective. OS, overall response rate, are key secondary efficacy endpoints, and they're kind of the standard. Safety tolerability, we'll be looking at some PK. We'll be looking at patient-reported outcomes and immunogenicity of the antibodies you might expect. In terms of exploratory endpoints, we'll be looking at endocrine expression on our angiosarcoma tumor specimens. Some have higher expression than others. Maybe that will correlate with activity. We look at circulating tumor biomarkers to see if there's something that might be predictive of activity, and also circulating tumor cells to see if that also might predict activity, which is exploratory analysis. So the way this study works, it is an adaptive design, and the initial analysis of interim analysis we expect in the second half of 2018. We're looking to see, we're basing our current, so if you look at our current data, we have 3.0 months as expected PFS for pazopanib. We've got 5.5 and a small sample size of even previous pazopanib treated patients. That's a cross-study hazard ratio of about 0.53, but it's across studies. And I'd argue the patients that got the combination of those 9 patients were pretty heavily treated because they'd already seen Votrient before, whereas the 3.8 month Votrient data, they hadn't seen it before. So it may be conservative, but still, we have limited data on what to say, what's the hazard ratio we would expect to see with this drug. We're powering the study at 0.55 as a baseline consideration. But obviously, we'd be incredibly disappointed if we achieved the hazard ratio, say, of 0.6 and missed the study endpoint with 124 patients. That would be a tragedy in our opinion, right? So we wanted to inform ourselves based on an interim analysis. And we're planning to do that at 70 patients at 40 events and do a conditional power assessment of the overall population, but also look at the cutaneous population. So 2 conditional power calculations at the interim analysis, and that will drive the final study. So we're putting a lot of faith in those first 70 patients, which, there's risk there. But it's better faith than what we have in terms of the available literature when you're in this orphan indication where you can't fully inform yourselves of what to expect, both in the control and the test group. So based on those 2 conditional probabilities, we will allocate the study into one of 4 zones. Okay? The favorable zone will be selected if there's conditional probability indicating a nice separation of the 2 treatment groups. And if we're seeing a nice separation, and by nice separation, I mean the conditional power in the full group is about 95% or higher, we would stick with the favorable zone, which means the study would stay unaltered. Now you can see we set that bar very high. We don't mind enrolling 200 patients versus 124. What we don't want to do is enroll 124 patients and miss the endpoint. The promising zone is, as you'll see, probably a more likely alternative given the conditional probabilities, or given the expected hazard ratio. And what the favorable zone does is it increases the sample size to 200 patients. What it's really doing from a global view is if you're not quite seeing the great separation, say from 4 to 7 months, say a separation of 4 to 6 months, that's important. If you can go to 4 to 6 months, our view, that's a very successful trial. You increase PFS in a very deadly disease by 50%. That's a good endpoint for us. And if we need to get 200 patients in the trial to see that, we have no problem with that. So the promising zone has a conditional probability roughly between 30 and 95, 0.3 and 0.95. So we don't mind selecting that zone if that's what the data dictates, and additional patients enrolling. By that time, you're starting to enroll pretty aggressively anyway. Another 6 months of enrollment to get a drug approved, in our view, is a small price to pay.
Where did the 200 come from?
So that came out of what we think is practical in a set time period of accrual, given angiosarcoma is a fairly rare disease. It also played a little bit into the fact that, what's clinically meaningful? If we can go 4 to 6 months, 2-month improvement, I think that's clinically meaningful. If it's 4 to 5 months, let's say we went to 500 patients for sake of argument, we could detect an improvement from 4 to say 5 months, just as an example. Is that really clinically meaningful? Not sure. So there were 2 considerations. What's clinically meaningful, but also what's practical in terms of accrual of patients, given this is a rare disease. And I'd say probably the latter was the most influential. We think we can still enroll 200 patients in this trial in about a 2-year period of time. So the enrichment zone is to try to take into account the fact, what if the drug's working in cutaneous patients very well, but it's not working in non-cutaneous disease? And in that case, we would only enroll additional patients with cutaneous disease, another 100 patients as an example. There would be 70 patients at the interim analysis, roughly half would already have cutaneous disease, and you'd enroll another 100 patients thereafter, so roughly you have 135 total cutaneous patients, which should allow you to still have the power to detect the original hazard ratio in cutaneous only. The unfavorable zone is when the drug, frankly, isn't working. It's not working in the overall population, and it's not working in the cutaneous population. By the rule of the study, you would still fully enroll the trial, because it's still possible the interim data underpredicted the real activity of the drug. But clearly, if the DMC felt that truly this study is futile, they could stop the trial at their election, but there's no set futility rule. Okay.
So
Please, yeah, questions, please.
The assumption of control is the 4 month, right?
4 months is our assumption for
But
control.
you have something coming from first line with the.
So we don't have great data first line.
That's for that, right? So,
We don't have, that's a great question. We don't have great data for first-line PFS. What we do know, though, is chemotherapy, and granted, it's a different mechanism of action is out there. Chemotherapy, whether it's first or second-line angiosarcoma, is about the same in terms of efficacy. So we feel like Pazopanib will probably be pretty similar. And there have been patients treated with Pazopanib first-line in angiosarcoma. For instance, even in that study of the 30-patient study, that top line of that graph, which is now 40 patients, about 14 of those patients came first-line in, and the PFS wasn't that difference. Now you see, we did take 4, not 3, to give some flexibility for maybe first-line patients do a little bit better. So we use those as the kind of basis for making 4 instead of 3. That it could be a little better, but it shouldn't be dramatically better based on how chemo performs and based on the limited data with Pazopanib first line. Yeah, great point.
Charles, I have
Please.
a question.
Yeah.
So the entry criteria is patients older than 12 years of age, right?
Right.
However, at the first interim, you're just including the adults, if I understand,
That's
by
right.
the time of the first interim. So when you calculate conditional power, you're assuming that the effect is basically going to be maintained for the final analysis. Is there a concern that patients could respond different, pediatric patients
Yeah,
could respond differently?
It's a great question, too. I didn't go into that level of detail. So the disease is rare overall. It's really rare in children, too. So what the EMA wanted us to do, which is what we did, so we actually, the pediatric patients 12 to 17 can enroll. They're all going to receive ampazopin, so they're not going to be actually part of the intent to treat analysis.
Oh, okay.
And that was really EMA wanting to give kids the best chance they have. So great question, and I didn't go into that level of detail, but that's why there won't be an issue with strict intent to treat analysis. Question, please.
So it looks like you have two decisions for favorism and unfavorable. However, the recommendation is continue the study as planned, 124 patients. So what has changed regarding, what's the perspective has changed regarding the two zooms, if you go to the next slide? Yeah, so.
The study will still say 124 patients. Absolutely, yeah. So, I mean, yeah, so
The details, like what.
nothing would change. Nothing would change. You would continue to enroll the study as planned. So we don't have a futility rule set, but the DMC, if they look at that data and say, there's no way that's going to work, so the probability is literally 0, they could stop the study at their election. But it's not programmed futility. So there's no rule for futility. Does that make sense? So yeah, the favorable and unfavorable zones are the same design. Just they could stop the futility in the unfavorable zone at the discretion of the DMC. Question, please.
I have a question about the endpoint. You really don't know the probability of free survival you will get, but you are getting complete responses. And complete responses are obviously the great breakthrough for the therapy. So did you discuss with the EMA and the FDA the possibility to use that as a probable endpoint?
Yeah, I think, so you mean like a single arm trial?
Or even randomized, but obviously a complete response, the noise level you have is 0 because
You do.
Yeah.
Yeah, we saw 2 of 9 complete responses. I think it would be risky to try to take that on. I think the nice thing about it is they were so durable that if we're seeing complete responses that are durable, that's going to play very nicely into our overall PFS as well. But to answer your question, I think the issue was, we couldn't count on that kind of activity. In a randomized study, I think we would get more power with the time to event endpoint, was our thought. We discussed, could we do it as a single arm trial? The issue there was, it's a combination therapy, so the agency was never going to say, oh, you can get approved on a combination therapy based on response rate, unless it was maybe 100%. So we did think about it, but I think we thought a time to event endpoint was more standard, and I think our available data supported that as a primary endpoint. Yeah, good question. So this slide's a little complicated. I just wanted to, so we really had a choice. We could do this adaptive enrichment design, or we could just compare that to a fixed sample size design. And I thought I'd just take you through a couple of the so Cytel did a lot of simulations around different conditional probabilities, and that helped us pick the borders. But just I thought I'd take you through kind of what it actually looks like practically, because it's not always win-win, obviously, right? If we did a fixed sample size design, it would take longer to do the study, but we'd have more power in most cases. The exception being if we do see that disparity between cutaneous and non-cutaneous. But let's just look, for example, at the top of the screen here. I'm going to go over here so I can read it. Oh, I can't read it there. So this is assuming a hazard ratio of 0.55 for both cutaneous and non-cutaneous, which is shown in the far left column. And let's just look at that first box going across. This is your idea of what we're giving up and what we're gaining. Now, if we look at the orange bar, that a priori gives us the overall power, 84%, to detect the endpoint. Okay? Now, that's the adaptive enrichment column. Okay? So just looking at the orange column, everyone sees 84%. So that's everything that could happen in terms of allocation to different 4 zones, but the overall power is 84% to detect the overall treatment effect, assuming that we have a real 0.55 hazard ratio in both cutaneous and non-cutaneous groups. Okay? Now, if we had done a fixed sample size, go to 200 patients, we'd have much more power, right? Because in this case, the adaptive design doesn't help us that much, right? It shortens the study, that's a good thing, but it doesn't help us too much in terms of the power because we're assuming the groups have the identical hazard ratio, right? So obviously, the bigger the trial, the better the power. If you have the hazard ratio the same in the two individual subgroups, right? And you can see within the kind of boxes above that orange line is, you can see though, where you get allocated based on that interim analysis is going to have a big impact on your overall power. Like if you have a true effect size of 0.55, but you are allocated based on just samples, just random responses or random PFS into the unfavorable zone, it's unlikely, your power is going to be very, very low. But if you're allocated, which more likely you would be, to the promising unfavorable zone, you have about 80% chance of being in those zones, your power is going to be much higher, well over 90%. So, allocation to the initial zone is important in terms of understanding how the study plays out, and you could be allocated in an unfortunate way, which would be obviously very unfortunate for us in the drug if it really worked, but we didn't achieve the endpoint because we were allocated because of kind of the random chance effect, if you will. Let's now contrast that, but let me just finish that thought on the study duration. The other advantage is the study duration is going to be shorter unless you go sample size re-estimation at 200 patients versus the fixed sample size design. The other thing that helps us as a small company is the interim analysis, people will know what the study's doing. And that helps us as a small company. If it's a fixed design, you don't know really what's happening for 2 years. We're going to know what's happening next year. If we're in the favorable and promising zone, that's going to be very valuable for us as a company. It's going to really help maintain enthusiasm of the investigators as well to keep this trial moving forward. So it actually has some non-statistical benefit to the company as well. Yeah, question, Nini, yeah.
So the DMC is the one who's going to...
The DMC will make the decision based on the conditional probabilities.
They will, say continue or not.
Continue, and they'll tell us what sample number to go to. Yep.
Okay, but if they don't say anything, you could be just as well in the unfavorable. So
You could.
You
could, that's true.
I guess,
That's true.
my question is, would they actually communicate that?
Yeah, we haven't discussed that in detail. That'll be discussed at the first charter meeting. It's a great point. Yeah, we'll have to discuss what the final sample size is because the protocol will be, will reflect that. But
It's an open label study, right?
it's open label.
So you can tell their look right results if you want to.
We can't. So this is all done through Cytel. So this is all through the DMC. So yeah, as a sponsor, we don't have any idea. Yeah, we won't be able to look at the data. Yeah, so all come through Cytel and the blinded reviewers. So yeah, question.
I was hoping to see a little more detail of how did you choose the promising zone and enrichment zone?
Yeah, we did a lot of simulations. So we did a lot of conditional probabilities. We did higher than 30. So the promising zone is between 30 and 95%. We closed that bracket a little bit more. Probably when you close that bracket, you might be more likely to stick in the favorable zone, which is a shorter study. But you also are going to be in that risk factor where you might have had a positive trial of 200 patients, but it would be a negative trial of 124 because say the hazard ratio is 0.6 as an argument. You may not hit it at 124, you would have hit it at 200. So in general, we made it wide enough that we wanted to be in the favorable or promising zone in about 80% of the cases at least. And if we narrow that bracket down, we wouldn't have. If you broaden it out all the way, you might as well just do a fixed sample size design, right? So it was a balance done with multiple simulations to kind of empirically determine those conditional power boundaries.
Even though this is done by DMC, because you are going to have a sample size increase decision, you still need to consider the type of error, right? Does that...
So type 1 error, I'll talk about in a second, but it does preserve type 1 error because even though you're using the interim analysis to inform the final sample size, you're not using it as a way to modify, to stop the study early. So in that sense, you preserve that error. Now, there are complex testing hypotheses for this closed test principle. I'm going to try to explain that, but I'm going to fail. So, Paul, you can help me. But to answer your question, it was empirically determined through a lot of simulations to try to put us in the favorable promising zone in most cases, but still get the benefits of a potential shorter study and potentially the benefit of enrichment, which I'll talk about in a second. Okay. Question, please.
Yeah, so if I understand, the sample size either stays the same or goes to 200, but nothing in between.
Well, it can go to 170. Let me go back on the slide, sorry.
Well, with the enrichment.
Yeah.
Let's forget about that.
Okay, sure.
Just to make it simple.
Sure.
So but it won't go anywhere in between that, right?
That's right.
So your conditional power could be 94%, you're in the promising zone, you're still going to increase to 200, is
It
that
could.
correct?
It's true.
All right.
It could. Yep, you're right. You're absolutely right. Yep. And then, yeah, and that was the, yeah, you have to, you want to give yourself a chance that if it's working really well, you just get away with 124 patient study, but, the tragedy for us was that it was, not quite going to work 124 patients, we work 200, that, this will be the only trial ever done in angiosarcoma. If we don't do it right and it fails, we're not going to do it at all. So, we're willing to take that hit to go to 200, just because we didn't want to pay the price of just missing it at 124.
I suppose this design has been discussed and agreed with agencies.
Mm-hmm. Yeah, it was amazing how it was not a ton of discussion around this. I think they really, they liked it. I mean, it's just put it out there. I think they really liked the design. Yeah.
Just ask a question.
No question, sorry.
So as a sponsor, how much guidance do you give the DMC about making the assessment in terms of allocating it to these various categories?
Yeah, that's a good question. And I think we'll have to discuss that at the charter meeting. It's a great question. My overview is, if it's unclear, like what if you're at 0.94? So those are the general boundaries, right? If you're at 0.94, the DMC can say, we still want 124 patients, right? So, it's not a firm rule, to your point. So there's definitely, they will make that decision based on the totality of the data, so to speak. So maybe I'll just look at one more category where the adapt design could really help the study. So let's just go back down to the bottom of the slide. So there, you can see, what's done here is that the hazard ratios are vary between cutaneous, shown on the left column, leftmost column, and then non-cutaneous on the second to the leftmost column. But if you look, for instance, at 0.55 and 1, so it's 1, 2, 3, 4, the fifth row down. So what if we're hitting the hazard ratio in cutaneous, but we just don't work in non-cutaneous? We'd still like to have a drug approved for cutaneous angiosarcoma. It'd still be a major breakthrough for those patients. So you can see there, again, this is the fifth row down. You look at the orange bar, with respect to the adaptive enrichment and the power, it's not great power. It's 60%, but it's some power. It's better than a fixed sample size of 51% if you just did a fixed 200 patient sample design. When you look at the individual categories, once the interim is done, you can see the enrichment zone could be picked in only a quarter of those cases. Unfavorable would be about another quarter, and then promising and favorable would be about the bulk of it. So you still have a good chance of going to 200 patients anyway. But you have about a quarter chance of going to the enrichment zone, which actually gives you the best chance of showing a treatment effect if the true hazard ratio is, it works in cutaneous and it doesn't work in non-cutaneous. And then the overall accrual period would likely be less than if you'd done the fixed sample design. So that's where you're actually getting a benefit of the adaptive design in terms of a power, not just the time, is in that situation. Will that happen? We're not sure. It could happen. That's why there's so much uncertainty here. But that's where the benefit really is across the board. Shorter trial potentially, better power to detect the positive effect than even a fixed sample design in 200 patients. So that's, so I'm picking the one example where it really shows the adaptive design is helpful. You can see there are other ones where it's close, but most cases, aside from that, the 200 patient fixed sample size will be superior. Again, we talked about the other advantages of the adaptive design. You get some information along the way that helps you maintain enthusiasm for the trials that moves forward. So, so I think I just kind of contrast two extremes. Identical hazard ratio 0.55 and 0.55 in cutaneous and nothing, no effect in non-cutaneous, where things can be allocated and what the overall effect is. So, type 1 error control came up. So, again, I'm not a statistician, but there are two key considerations. I didn't go into this in great detail. So, you're going to enroll 70 patients roughly in what's called cohort 1 to do the interim analysis. Now, cohort 1 and cohort 2 have to maintain their independence to do the test adequately. So, in 70 patients, we're expecting 40 events at the interim. We then want to get 60 for the full analysis, and then that's cohort 1, which contributes to the overall statistical calculations. You know, one thing we are doing, if we don't hit 60 events in cohort 1, we can actually increase cohort 1 by 20% of patients to get those events, because it's critical to hit the events, obviously, to get the endpoint. As long as we make that decision before the interim analysis, so just to be crystal clear, as we're enrolling cohort 1, we're going to look at the event tracking. If it's 70 patients, we're not quite where we think we're going to get 60 events of those 70 patients. We can increase to, say, 84 patients, so we'll get those 60 events, which preserves that power. We need the events to get the power. In cohort 2, we can also do the same thing. As we build out cohort 2, you know, for instance, even the fixed sample size, say it's the favorable zone. So we're only approved 54 more patients in cohort 2. We have a certain number of events we want in that group, and so I can't remember exactly what it is, but we can increase cohort 2 another 20% to get those events as well. So as long as we make those decisions, cohort 1, increase the size before we start cohort 2, it's okay, and we do cohort 2 independent because cohort 1 is already finished. And so you maintain the type 1 error by maintaining the independence of cohort 1 and cohort 2. And you also maintain the independence through testing both in the full population. For example, you're in the enrichment zone, you're going to test the full population and the subpopulation, and you've got to have statistical significance across both those populations to reject the null hypothesis and to show activity in that cutaneous population. So the two key things are the interim analysis doesn't modify the trial in terms of allowed endpoint determination, just resample sizes the trial. Cohort 1 and cohort 2 remain independent, and with doing those two considerations, we can then use this closed testing principle of Jenkins et al. We can maintain the ability to test statistical significance, whether it's whatever the design is, whether it's in the 124, 170, or 200 patient sample size at the end. So I know that's may not be crystal clear, and you may know more of those closed testing principles than I do, I'm sure. But